granular gases and biophysics and non-statistical physics and life.
Thank you very much for the introduction. Good morning. There are many places called Auckland all around the world, from California to New Zealand, but there is only one Auckland University, which is in Michigan, and near Detroit and Ann Arbor. Well, so in contrast to earlier talks in this session, I will not be looking inside the cell. So my basic object is a single cell, about 10 microns in diameter, and I would like to describe collective behavior of huge number of those cells. I collaborate with the experimental group of Mike Chok from at Henry Ford Hospital, and this work was done with Len Sander, who was my postdoctoral advisor, and with two experimental groups in Ohio and Arizona. So first, I will present some biological background, and then I will describe our discrete modeling approach and study the effect of cell-cell adhesion on clustering. And probably I will not have time to mention another approach, which is a continuum one, so I mention it now. So I am dealing with glioma cells. So the diameter of glioma cells is about 10 microns. So what those glioma cells can do? So here you can see an experimental snapshot, and every green dot here is a cell. So what those cells do? Those cells randomly migrate when you put this on a substrate. They can randomly migrate all around the substrate. They proliferate. They may form small clusters due to cell-cell adhesion. Now, the typical time for migration is rather small. So a cell migrates its own diameter, 10 microns, in about 5 minutes. The typical time for proliferation is much larger. It may depend on different things, but order of magnitude is 1 day. And then cells form those clusters, so those clusters can be, so a cell can attach to the cluster and detach from the cluster and migrate away. And now I would like to say a few words about this kind of tumor. So glioblastoma multiforme is one of the most dangerous and malignant form of primary brain tumors. If you Google glioblastoma multiforme, you find much more than 700,000 links. And here I listed several unpleasant facts about this awful disease. So in general, the treatment is highly unsuccessful. And one of the main reasons for this is the fact that those tumors are highly invasive. It means that cells not only proliferate, not only divide, and then the tumor grows like this by proliferation, but cells also detach from the primary tumor and migrate away into the extracellular matrix. So actively detach and migrate away into the matrix. And so one can detect those tumors and hopefully remove them. However, many cells are left out, in the many cancerous cells are left out. And this is the main problem. So in order to study these, people perform experiments in vitro. In those experiments, for example, one can put a small tumor spheroid of about 500 microns in diameter, put those spheroids in transparent gel, for example, collagen gel, and just follow the dynamics of growth. So see how does it grow. You can see that the typical tumor consists of two zones. There is an inner core which grows with time, and there is outer zone. So every black dot here is a cell. So cells detach from the primary tumor and migrate away. So, and as I said, you can detect and remove the primary tumor. However, there are many invasive cells which are left out. Now, interestingly, quite amazingly, the cells on the surface of this tumor and individual cells which migrated away, they do not behave the same. So biologists say that they have different phenotypes. So those cells have proliferative phenotype, and those cells have invasive phenotype. This means that the proliferation rate, the rate of division of those cells is much larger. And those invasive cells, they just they move, and their proliferation rate is very low. Now, well, one can ask, so if those cells, those invasive cells do not proliferate, so why it is bad? So suppose you take out the primary tumor and you have a bunch of cells, and if they do not proliferate, maybe it's not so bad. And the answer is that at some stage, it is not known why or how does it happen, but at some stage they switch back their phenotype. So they stop and they start proliferating. And that's how secondary recurrent tumors are formed, and this is basically one of the main problems here. So the question, of course, one of the main questions here is why those invasive cells switch their phenotype. So what is the mechanism which govern this switching? And I will try to say a few words about this. So there are many different cell lines, glioma cell lines. For example, there are those cell lines, I will call them wild type, and there are many mutants. One of the mutants is when there is an amplification of some gene, of growth factor receptor gene, and if you take those spheroids and follow their dynamics, you can see that the pattern is somewhat different, and there are many differences here, but I would like to focus on those small clusters. Yeah, those are just small cell clusters which are formed in the invasive zone, and maybe those small clusters will develop into the secondary tumors, afterwards when the primary tumor will be taken out. So the question is, why those cells cluster? So let me present our discrete agent-based model. So consider it, this is a two-dimensional model. So consider a lattice, and every lattice side can be occupied by a cell or can be empty. So cells so the dynamics is the following. You pick a cell randomly, and then a cell can proliferate with some probability alpha, or the cell can migrate to a neighboring site with some probability 1 minus alpha. So as I said, the diffusion time is much shorter than the time for proliferation, so alpha is a small number. Now, this doesn't take into account cell-cell adhesion. In order to take into account cell-cell adhesion, I want to say that this cell, which has many neighbors, it is rather hard for it to migrate away because it needs to break bonds, compared to this cell that can freely migrate, freely move. So, in order to model this, I introduce adhesion parameter Q, and I say that the probability for proliferation is alpha, and the probability for diffusion is 1 minus alpha as before, times 1 minus q to the power n, where n is the number of nearest neighbors. So if q is 0, no adhesion. We are back to the previous case. Now, if n is 0, so there are no neighbors, again, the probability for diffusion is just 1 minus alpha. However, if both q is larger than 0 and n is larger than 0, then you can see that the probability for migration decreases with the number of nearest neighbors. Now, quite amazingly, this model in some cases can be mapped into the Ising model in statistical mechanics. So let us, for a moment, turn off proliferation. Yeah, the proliferation is small. It is there, but it is small. For a moment, we turn it off. Now, if we turn off proliferation, then you can see that a mapping goes as following. An empty side corresponds to spin down. An occupied side corresponds to spin up. And then the magnetization corresponds to the average density, and the adhesion parameter corresponds to the ratio between magnetic coupling and temperature. So we can use this mapping and say that instead of plotting magnetization temperature phase diagram, we can plot adhesion average density phase diagram. And on this phase diagram, there are two qualitatively different regions. So below the critical value of adhesion, the homogeneous state remains homogeneous with time. However, above the critical rate of adhesion, large clusters, there is phase separation, and large clusters grow just by coursing. However, of course, there is proliferation. Proliferation is not 0. So let us turn on proliferation. If we turn on proliferation, the average density of cells increases. If the average density of cells increases, depending on your adhesion parameter, you can either enter the unstable region or remain outside. If you enter the unstable region, you get clusters. If you remain outside, the system is homogeneous. And then I convinced our experimental collaborators to perform experiments with wild-type cells and mutant-type cells we saw before. So they just put those cells on Petri dishes and just saw what happens. So you can see that mutant-type cells form clusters, but wild-type cells remain homogeneous, which suggests that those cells have effective adhesion which is above some critical ratio. To do this, to analyze those experiments quantitatively, I measured cluster size distribution function. So I can look only on the tail of this cluster size distribution because we cannot detect very small clusters. Now, if I compare this with experiments, the comparison is not so good. If I compare this experimental curve with simulation, the comparison is not so good. So in simulations, you have large number of almost equal size clusters, where in experiments, number of clusters is smaller and there are differences. And probably this is because we didn't take into account another mechanism. So there's one mechanism, and another mechanism is what I mentioned before. So cells on borders, on surface of the cluster, they proliferate more than individual cells which just freely migrate. And if I take this into account in my discrete modeling, saying that there is some basic proliferation rate, but cells on this surface proliferate faster, then the agreement between simulations and experiment is much better. So this is the experiment and those are simulations. So and so you can find parameters both for wild type cells which remain homogeneous and mutant type cells which cluster. So in conclusion, I would like to say that there are two scenarios for time evolution and growth of cell population. So if the adhesion is rather relatively small, the system remains homogeneous. However, if the adhesion is above some critical value, there is safe phase separation between high density clusters and low density cells, individual cells, And on top of this, once those small cell clusters are formed by adhesion, cells in those clusters start switching their phenotype and may start proliferating more, which leads to a rapid growth of this cluster. So we can guess that those mechanisms have something to do with the formation of secondary tumors. Thank you for your attention.
Any questions?
Hi. It's not clear to me that the density is what's driving the increased proliferation, because you have a mutation in those cells which is causing the growth, the EGFR to constantly signal. So it seems like you need to do some kind of control experiment where you have the same mutation but then prevent the clustering to show that it's really density dependent.
Actually, it was my first, thought that it is density dependent, but if you perform an experiment just in homogeneous, so put cells homogeneously, and cells grow with time, and then the density changes, and you can follow, cells you have. So in this situation, in a homogeneous situation, you can see that there is no density dependence at large. So when cells are almost densely packed, the proliferation decreases as inside the tumor. However, for large intervals in density, there is no density dependence. What I'm saying here is slightly different. I say that cells on the surface of a tumor proliferate more than individual cells, and this is experimentally known, in gliomas, both for mutant and for wild type. That cells, most of the proliferation occurs in the surface. So here inside the tumor, cells are so densely packed that they cannot proliferate. However, and cells here proliferate rather fast, but those individual cells, their proliferation is by order of magnitude lower.
Hi. Yeah, it's on. Oh, it's on.
Hi. This is probably a sort of naive genetics question, but I can physics background. How is it that you have a wild type and a mutant of a cancer cell? Because in my mind, a cancer cell occurs by mutations of a healthy cell.
No, it's not that you compare healthy cells and cancer cells. There are many types of different cancer cells, and so those are not mutations which lead to cancer. Those are other mutations. You can induce many mutations, and there are many cancer cell lines, huge number of.
So for this cancer cell you're studying, is it a specific mutation that leads to that cancer cell?
Well, I don't know what, and I'm not sure, if it's well known what mutations in healthy cells lead, to those cancer cells. But you can play with different cell types of cancer cells. So those are not mutations which distinguish between healthy and cancer. Both of those cell lines, U87 wild type and U87 delta EGFR, both of them are cancer, and it seems that delta EGFR is even more aggressive, and maybe this is because they have an enhanced ability to form those clusters.
Any other questions? Okay. If not, I think we're going to start our panel discussion, if you will. So if will the other speakers please come down.